So I'm going to tell you a little bit about a class that I've been teaching at UCI. Some people elsewhere have stumbled across it already. So it's called Computing Techniques in Drug Discovery. But before I do that, I'm actually going to tell you about something else that relates to the topic of this forum that people might be interested in, another educational resource. There's a journal we started a while back called the Living Journal of Computational Molecular Science. And I just kind of want to point people to it since it's not the main point of my talk, but it is. So that's where you find it online. It's the thing that's interesting about it that might interest this audience is it's particularly focused on educational things and things that require updating. And so those include like best practices articles or tutorials or review papers that you might want to keep updated. And I'll give you a couple examples of that and how they might help. But think especially of the tutorials and best practices. So we had a couple issues out in 2019 already, and there's another on the way. So here's a couple of several of the first few articles. There's some tutorials on a method for sampling rare events with molecular simulations. There's some GROMACS tutorials. There's some tutorials for Markov state modeling. There's some things on how to simulate lipid membranes, or some basics on molecular simulations or computing transport properties, or uncertainty quantification for molecular simulation. So all that stuff is online, and one of the other interesting things about it is it's all CC BY content. So basically you can reuse anything from it for any purpose as long as you credit the authors. And authors don't sign over copyright to us to the journal when they do this. So we just wanted to point it out in case it's either something you might benefit from in your own, being able to use this in your own work, or in case you do work and you might want to consider this as a place to publish these types of articles in the future.
And David, we can, if you're willing to give us your slides, we can put that on and make it available to everyone. We're only seeing your main slides. I don't know if you were showing something, but yeah.
It should be these main slides, like with the titles of articles on it.
All I see is teaching computational chemistry.
Oh, it's not advancing? No, that's not good.
No.
That's interesting.
Okay, now we got it.
Well, okay. So I tried to show this.
Yeah. Okay, good.
And I tried to show this with some, I don't know why I wasn't doing that before, has several different titles of articles on it, and then a third one which has other titles of articles on it. But okay, I'll show the slides later.
Yeah.
So to get on to the main thing, so the class I've been teaching at UCI, it's a semi-flipped course. So this is basically a class that I teach that is about, well, it's sort of the class for students who are going to be in my area or should know something about my area of like computer modeling at the interface of, at the edge of drug discovery, basically. And so I wanted to make sure that I was actually teaching students how to do something useful in the class, something they might actually use in their research, rather than just giving them like sort of theory and concepts and stuff. So it's a graduate level course, although some undergraduates can take it also. And it's semi-flipped. Perhaps 2/3 of the classes have a hands-on component. And in those, what I do is I'll give them the slides ahead of time. Actually, I always have the slides up ahead of time. I encourage them to read the slides in advance, and then I'll go just through highlights and then spend time in class helping them work through some problem solving and testing out the stuff that we're learning. And sometimes that includes, there's a pretty big project component to the course. They do sort of mini projects in the context of the course. And so we'll spend some in-class time working together on that as well. The idea being, I figure students get a lot more actually useful stuff out of learning to do things along with the concepts rather than just learning concepts and then having to figure out the stuff on their own. I do require significant programming experience before people do it, but I start with a crash course in Python. And I cover a pretty broad swath of computational chemistry as I do this. The grading ends up being based on these mini projects that are at almost a research level. The idea being that I want them to be able to take what they did for these mini projects and take it back to their labs or their research and actually do research projects with it. And I'll show you some examples of the content in a couple minutes. So all this is up on GitHub. Here's sort of a screenshot of where it is, and you'll have the URL when I share the slides later. So it has a bunch of different, some documentation on installing stuff. There's a syllabus. There's some assignments that I give. The lecture content is there, and the assignments are up as well. So I'm going to switch over and actually show you some of the content. I've sort of singled out a couple examples that I might show you. One is dealing with working with molecules and shape. Another on fluctuations and correlations and error analysis, and that deals with some sample data from molecular dynamics simulations. And something on host guest simulations, but we'll see how far we go through those. Let me stop sharing that screen. And so these are Jupyter notebooks, which is a Python thing that some people will know. And so those are gonna run in the web browser.
Okay, are you
so hopefully you can see that now?
Yes,
all right.
we can.
So there's a notebook here that looks kind of small. If I do this in a fancy way, I can switch it to slides. So does this look nearly full screen now?
Yes, it does.
Okay. So it starts off with this blurb. Then this I could project it on an overhead. And so it has some content about what we do with work with molecules. This is very early in the course, how you might start with working with molecules from a smile string or from a 2D or a 3D structure. Has a bit about how to load them. And then in the course, we rely quite a bit on the OpenEye toolkits. So I do some intro to that. The cool thing about this, though, is that you can actually run Python in the notebook. So I'll show you more of that in a minute. So I'm skipping through some of the content. It shows how you might, as a starting point, you might come in with a 2D structure of a molecule you've already drawn. And so then we're going to go ahead and load a molecule into Python. So I loaded this one, but we could switch the smile string here to something else and load a different molecule. And this will get a little fancier. So, and you can actually draw that same molecule in the notebook. I just drew it. So the students could, go back to the previous slide, put in a different smile string, redraw a different molecule here. So there's an opportunity for them to play around with that a little bit. Then you can, put in your favorite name of a molecule and do the same thing. You can draw that. So this is that same molecule, but we could do, let's just do phenol here. And there we have it. So you're running Python code in the Jupyter notebook on slides, which I really like. And so it gives them a chance to play around with it and see how it works. And we can do the same from a file. And then there's also this isn't a super important example, but it shows them how to render PDFs of their molecules. So there's sort of a mix of Python code and informational content here. Shows them how to loop over some properties of their molecule. And so this is kind of boring, but I show them how to do a for loop where they loop over the atoms in their molecule, and you'll see where this is going in a minute. Let's see. Okay, I should run that so that I have the correct. Right, okay, so I did that. 1 sec. Whoops. I've forgotten to run one of my boxes. I'm gonna flip back through here and see where I forgot to run my box. Oh, here we go, missed this box. Okay, so I'm generating a couple more molecules, writing them out to files. Now this is something I like. You can actually, so we've generated a 3D structure of this molecule, and I'm not going through all the content with you because you're not in my class, but I showed people how to, some of the code shows how to generate a 3D structure of a molecule. And so now I can take the 3D structure that I generated and actually spin it around in the Jupyter notebook, and they can switch which molecule they've done this on. So where all of this is going is partly I wanted to show them the idea of like shape comparisons of different molecules. And so there's a literature example of doing an overlay of morphine and tramadol, which are pretty different 2D structures, but once you put them in 3D, you can see that they can occupy the same shape in a binding site. So we can do that here. So we pull morphine, pull tramadol, except it doesn't understand the name. That's tough. So Wikipedia saves the day. We can pull the IUPAC name for that, and it does understand that. And then I have the students use the examples that I put up above to write code, and this is something they would do in class to visualize the molecules to make sure that they processed correctly. Then go ahead and do conformer generation for the two, and so get 3D structures for them. And let's keep going. Whoops, I missed that box. So since it's Python code, I have to remember to, whoops, I missed a box. Have to remember to run all of my boxes. And I think I still missed a box. Sometimes I forget the correct way to advance or not advance. Yeah, here we go, this box. Okay, so back to our slide view here, whoops. Okay, so now I've done a shape overlay of the two. This is basically reproducing the literature example.
And Carmen will tell you a lot more.
Occupying, and if I want to be fancy, I could color code them differently, but you can see these are occupying very similar shape, very similar space, which relates to the fact that they can bind to the same receptor. So that's the main thing I wanted to show you from this example. There's a bit more here, but you're not in my class, so I'm trying to just give you the highlights. I wanted to show this to you partly because I think some people find it useful. Sometimes people find the course useful to help students get an idea of how to do some of these basic tasks. Also because you're welcome to reuse it in any way you want. Again, it's under a permissive license, so you could do that. And finally, I'm happy to have contributions. There's another couple of notebooks I could show you if you want, but I know there's not that much time, so I wanted to make a couple of final points. One, not the only one doing things like this. To avoid switching back to my slides, I'll just explain. Andrea Volkamer's lab has a sort of similar set of contents that covers a bit different topics called Teach Open CAD. So you should be able to Google for Teach Open CAD. I think it's Teach Open and then CADD. And that's the Volkamer lab, Volkamer. And so theirs focuses on using fully open source tools. Mine, I tend to rely quite a bit in some parts of it on the OpenEye tools because I use those more. They are free for academics. So if you're an academic, you're certainly, it's an easy thing to get a license to, and they're very useful. So I'm happy to take a couple questions now. Couple of the other, but just to show you the other things that I'm not sure that I'll get through, but I have them up here in case anybody wants to know. One of them is a bit about setting up a more advanced one on setting up some simulations of host-guest binding. It actually runs a little molecular dynamics simulation in the notebook. And then I have one on fluctuations, correlations, and error analysis that has a bunch of sort of background content in it, and then it actually takes a sample simulation and tries to detect whether the simulation has run long enough and then continues running it until it's run long enough. But anyway, I'm happy to take some questions there or show people a little bit more if you want, either way, or be done.